In this lesson, we apply Mendel's principles by predicting the outcomes of inheritance using probability. All right, so this lesson is about applying some of those principles that we learned that Mendel had figured out in his experiments. Now, as we get into this, we're going to kind of take Mendel's principles and apply them in ways that he was never able to because he didn't have the technology to see what was going on in some more complex arrangements of alleles. But we're going to get into that. So let's go ahead and get started. All right, so we're going to talk first about probability and how that pertains to heredity. But first, let me ask you this. Suppose you toss a coin, right? Bloop. What's the probability of the coin landing on tails? And of course, the opposite question is what is the probability of the coin landing on heads? Well, if you said it's a 50% chance of landing on tails, or a 50% chance for heads, or half, you would be correct. That is absolutely, absolutely correct. So it's 50% or it's half, right? Now, what about if you tossed it 3 times? What would happen then? Well, there is a ratio of averages that tell us that 1 out of 8 times you would get tails, if that's what you call. The way that they figure that out is they take 1/2, because that's our probability the first go around here, and they multiply that 3 times because you toss the coin 3 times. That gives us a probability of about 1/8. So probability is something that we can use to predict what the outcome could be, but you never know until it actually happens. Okay, so these are predictions, and predictions can be wrong. Okay, just so keep that in mind as we go along here. Now, both the F1 plants in this particular scenario have the same set of alleles, and they are tall. Now, in the cross we have here, we have the big G, and big G, little g allele combinations produced in 3 of the green pea plants that you see here. So both of the F1 plants that we have here in this illustration have the same set of alleles, and they are also tall. Now, the cross we have here has big G, and big G, little g allele combinations produced in 3 of the green pea plants, while the little g allele combination produced the 1 yellow plant. So these quantities follow the laws of probability. All right, so let's kind of walk through this. So we have the F1 generation, and we see that the alleles for this green one here are G and G. Okay, this green one is also G and G. That's heterozygous, right? Because they have one of each, one big one little one, or one dominant, one recessive would be the technical term. And if we separate their gametes, their alleles, into the gametes, we get a big G, we get a little g, we get another big G, and we get another little g. So now we're going to cross them to see what kind of different combinations we might be able to come up with if these 2 parents have offspring. So we could get a big G and a big G, okay, which would result in a homozygous dominant, right? That's the allele combination here. Another possibility is big G, little g. So this big G and this little g could combine to give us another green pea pod. That would be heterozygous. We could get this big G and this little g. That's the same as this combination. So there's 2 out of 4 possibilities that we could get big G, little g. That's going to give us another heterozygous, which would be green because the dominant allele is present. Or we could get the little g from each of the parents and end up with a yellow pea pod. All right, so let's see. If we look at the first question here, the probability of a gamete receiving a big G. What is the probability of a gamete receiving a big G? So what we mean by that is if we separate the two alleles here, what are the chances the gamete's going to get a big G? Well, it can either get a big G or a little g, so that's going to be 50% or half. Okay? What's the probability of a gamete receiving a little g? Well, that's the same thing. There's only two options. It's either big G or little g. So that again is 50% as well. Now, the probability of once those alleles in the gametes combine, once two of them combine, one from each parent, what are we going to end up with? What is the probability of having green offspring? Well, if you count them up, 1, 2, 3 out of 4. So 3 out of 4, or a 75% chance if you convert that into a percentage. Because if you divide 3 by 4, you get 0.75. You multiply by 100 to make it a percentage, and you got 75. What is the probability of having a yellow offspring in this case, though? The only chance of that is a 1 out of 4 probability of having yellow offspring. So we're predicting the outcomes. We're not really saying, well, if these two plants combine, we're going to get a homozygous recessive offspring. That's only a chance. So there's a 1 in 4 chance that we're going to get a yellow offspring. So it's actually pretty unlikely because it's only a 1 out of 4 chance, or a 25% chance, right? 1 divided by 4 again is 0.25, multiplied by 100 to make it a percentage, it's 25%, okay? So we can predict, but we don't know for sure until we actually see the offspring. So there's a 50% chance that the offspring will be heterozygous, right? Because 2 out of 4 times you're going to get a heterozygous offspring. And you could also get a homozygous dominant plant, which would only be a 25% chance of that as well, just like the yellow. So you're probably going to get a heterozygous, or you're going to get either homozygous dominant or homozygous recessive. Those are the 3 possibilities: green, homozygous dominant, heterozygous, or homozygous recessive. Now, what are the 2 possible genotypes of a green plant? If they're green, what are the 2 possibilities? Well, the answer is they're either going to be big G or big G, little g, right? Big G, or big G, little g. So what is the phenotype of a plant that has 2 alleles for yellow color? The phenotype is yellow. Now, let's back up a little bit because we want to talk about these 2 terms, genotype and phenotype. So we have genotype. Genotype is the genetic makeup. In other words, those are the letters. So our genotype on this illustration would be this, genotype. I'm just going to put geno because they both end in the word type, right? See that there? All right, so this is the genotype up here. The phenotype is the actual outcome, the thing that we see. So this is phenotype. So what we have here is we can have 2 organisms that share the same phenotype, but yet they might have different genotypes, okay? In other words, both of these plants here, both of these offspring, they have a genotype of big G, little g. They're heterozygous. But their sister or brother, whatever this is, might have a big G, but still the trait we see, the phenotype is still green, but the genotype is different than the sibling. Okay? So phenotypes can be the same even though the genotypes might be different. Okay? Now, the yellow one doesn't share the phenotype or the genotype of any of the siblings. One way that I use to try to remember which one is which is the word physical trait starts with a PH and phenotype starts with a PH. So that's how I remember that phenotype is the physical traits. And the word gene, and genetic makeup, gene, both start with GEN. All right, now there's a couple other terms that I've been bandying about, been throwing around, been saying, and so you probably have a pretty decent idea of what they mean already. But let's just talk about the definitions real quick. First, homozygous means it has two identical alleles for a particular gene, whereas, and that's where we get the term, or the prefix, I should say, homo means same, right? Whereas hetero means it has two different alleles for a gene. And hetero, that prefix, means that it's different or other. Okay, hetero means other. So two different alleles for that particular gene. So which of these offspring here are homozygous? Well, the homozygous, they can be either uppercase or lowercase. So in this case, we get 1 and 4 are homozygous. So both of these are considered homozygous, even though they're not the same, but they both have the same allele for that gene. Okay, they both have either, in the case of offspring 1, we've got two dominant alleles. In the case of offspring 4, we have two recessive alleles. Now, the two offspring that are heterozygous are 2 and 3, obviously, because they have one of each. They have a dominant allele and a recessive allele. Now, one thing that we have to keep in mind is if I say, oh, that plant is homozygous, you will not be able to tell me whether they're homozygous dominant or homozygous recessive unless you know the phenotype and you know what trait I'm looking at, and you know the phenotype for that trait. So a lot of times, if we're just discussing genotypes and we're not shown the phenotype, we have to designate that it's homozygous dominant or homozygous recessive. So if it's a big G, it would be homozygous dominant. And if it's a little g, then it's homozygous recessive because it's only got the recessive alleles. All right, now there is a way that we can predict the outcome of particular crosses, and we do that by using a model that we call the Punnett square. Now, you may have encountered Punnett squares in the past, whether it be in a previous life science course you took, or in middle school, or maybe even you took a life science here at Achievement House. But the best way to predict the outcomes is to use these Punnett squares. So these are basically mathematical probabilities, and it just helps us predict what the genotype and therefore the phenotype combinations could be in a particular genetic cross. So the first thing we have to do is identify the alleles of each parent, the alleles that they could pass on to their offspring. And in the case of what we have on the board here, we're going to cross some ospreys that are both heterozygous. Okay, you should know what that means when I say they're both heterozygous. It means each will have one big B and one little b. We're talking about beak size, so we're just going to use the letter B for that. Okay, so then once we know the genotype of the parents, we have to separate their possible gametes, right? Separate their alleles. And that's what's happening here on step 2. We're figuring out the gametes. So here's parent 1, and here's parent 2. And the possible gametes that they can provide are either a big B or a little b because they're heterozygous. So we take one of the parents and we transmit them or transpose them to the top of the Punnett square. So we're just going to put parent 1 in the top, and then we're going to put parent 2 on the side. Okay, so each column gets one of the alleles from parent 1, and each row gets one of the alleles from parent 2. And then it's just a matter of combining them up. Okay, so we basically take the big B in this column, and we place a copy of it in each box. The result of our crosses is shown right here. But you see that we have big B in the first box, big B in the second box. Okay, so this is combining across and combining down, the intersections. Okay. Once we have the Punnett square filled in, we can now look at our ratios. So what this tells us is the boxes that have been colored in yellow are the probabilities, are the possibilities of having the phenotype of the dominant allele. Okay, because there is a dominant allele in each of those boxes, at least one. So if the dominant allele is present, then the dominant allele will show up in the phenotype. 3 out of 4 chance, right? There's 4 possibilities. 3 out of 4 times you're probably going to get the dominant phenotype because you can either get a homozygous dominant or a heterozygous. Both of those result in a dominant phenotype being expressed. Okay, there is a 1 in 4 chance that the recessive phenotype will be expressed because there is a possibility when you have 2 heterozygous parents that you could have a homozygous recessive offspring. 25% chance of that happening. That doesn't mean 25% of the time. That means there's a 25% chance each time an offspring is born. So we also know from this result that we could have either we could have 3 different genotypes, but we could only get 2 different phenotypes. But in a simple cross like this, in a what we call a one-factor cross, you see that up here? In a one-factor cross, you can only ever have 2 phenotypes anyway. But you could have up to 3 different genotypes. Sorry, you'd have 3 different genotypes, but only 2 phenotypes because it's a one-factor cross, meaning there's only one factor in determining the outcome. Okay, so we have a big B or a little b for each of the alleles. Okay. Now, it's possible to look at more than one characteristic at the same time, and we can still use a Punnett square to do some of those predictions. So when we focus on 2 different characteristics at the same time, we call that a dihybrid cross. The prefix di, di, means 2. So we're going to look at 2 characteristics in this case. We can also call it a 2-factor cross. And that kind of harkens back to what Mendel called the alleles. He called them, or the genes, he called them factors, right? So in a similar manner as in the previous illustration, we're going to kind of walk through this diagram, and I'll explain how to make the Punnett square for a dihybrid cross. It's basically like if you've ever played tic-tac-toe, but you played the complicated version of tic-tac-toe that has a lot more boxes. That's this, right? So with a 2-factor cross, again, we just start with the parents. So we take a cross of pea plants, and the 2 factors are size and pod color. Okay, so if we have 2 parents that are heterozygous for both factors, for both genes, that means we have a big T, big G, little g. Okay. We also have a big T, and a big G, little g for the other parent as well. So that's parent 1 and parent 2. So here's parent 1, here's parent 2. Okay. The possible gametes, these are the combinations that could end up in the eggs or the sperm of these parents. And so we need to get one letter or one factor, one allele from each of the two factors. So we need a T and a G in each gamete. So the possibilities that we can get from this are big T, big G, right? Because there's a big T here and a big G here. We could also get big T, little g, or we could get little g, we could get little t, big G, or we could get little t, little g. Those are the four possibilities if we were to mix up these alleles uh, into the gametes. So each gamete has to get an allele for size in this case and pod color. So we have to have one T and one G in each of the gametes. So what are the possible combinations that we could get here? This is going to we're going to get into a discussion here shortly about independent assortment. And that's really what's going on here. Each of the alleles gets assorted independently. So it's really random, but we end up with only one allele for each gene in each of the gametes. But because we're looking at two different genes, we have to have two different letters in each gamete instead of just one that we had before. Okay. So if we take these four possibilities from each of the parents, and obviously they're going to be the same because both parents have the exact same genotype, we're just going to put parent 1 across the top, so parent 1 here, and we're going to put parent 2 over here on the side. Okay, and they're exactly the same. But then we have to go through and write out our genotypes. Got to carry the big T all the way down this row and the big G all the way down this row. And then we have to take the big T from over here and put it all the way across this row. I'm sorry, I said row before. We have to take the big T and carry it all the way down this column. Take the big G and carry it all the way down this column as well. And then we do the rows. So we can go through each of the columns, just carry them. We write those two letters in each box going down the column. And then we do the same thing, but going across the rows using the gametes on the side. What we end up with is shown to us over here. These are the possible combinations. So basically, they have green depicting the possible genotypes or phenotypes that will be green. So this is 1, 2, 3, 4. 1, 2, 3, 4. 4 times 4 is 16. So there are 16 boxes in here. And what we see is we've got a 12 out of 16 chance of having a green offspring, having a green pod color. Now, they also used red font for the letters for the plants that would be tall. So if we count those up, we get 1, 2, 3, 4, 5, 6, 7, 8, 9. So it's 9 out of 16 chance that they'll be green and tall. Okay. There is a 3 out of 16 chance that they'll be green and short. Okay, you follow how this is working? For the yellow pods, there's only a 4 in 16 chance. That's basically 1 in 4. A 1 in 4 chance. Is that right? 4. Yeah. A 4 in 1 chance that a 1 in 4 chance. Sorry, a 1 in 4 chance that you're going to have yellow colored pods. But there's a 3 out of 16 chance that you'll have yellow. It's so confusing. You'll have yellow and tall, right? Because the text is red and the background of the box is yellow. So that would be tall and yellow. The black text with the yellow background would be short and yellow. Okay. Now, after Mendel showed that the alleles segregate during the formation of the gametes, he still was like wondering if the segregation of one pair of alleles affects another. And so to figure that out, what he did was he followed two different genes. He followed P color and P shape. And he followed them from one generation to the next, just like he was doing before. So we're going to use another dihybrid cross Punnett square because it involves two different traits. It involves seed shape and color. So what we know is that round is dominant over a wrinkled shape, and that yellow is actually dominant over green. We've been kind of looking at green as the dominant, but it's actually yellow that's dominant over green. So how is the genotype of the offspring different from that of the homozygous parent? So we see we have a parent up here that has all lowercase letters. So shape, let's see, which one is shape? I think shape is R, because for round, right? Round is the dominant allele, so we'll use the letter R. And yellow is the dominant allele for color, so we'll use the letter Y to represent the color alleles. So this parent is homozygous recessive. So HD for this parent. So the possible gametes that this parent can give is simply RY. Those are the only possibilities. When we have a homozygous dominant parent, which is big R, big R, big Y, the only possibility in the gametes is going to be big R and big Y. Okay? So for our first generation, our F1 generation, we end up with the dominant alleles for both color and shape being expressed because we get heterozygous results. The only possibility is a big R and a little r combining, and a big Y and a little y combining. Okay? All right, so what would the offspring be for the F2 generation? So the F2 generation would be taking two of the F1 generations and running a Punnett square for those. You would have to do a dihybrid cross Punnett square. All right. So if you look at this dihybrid cross Punnett square, can you find the homozygous recessive offspring? Well, if you found the little guy down here in the bottom corner, you would be correct. How many different phenotypes are present in the offspring of the F2 generation? Well, there are actually 4 different phenotypes. So what is the probability of a cross of these parents producing a fully homozygous recessive offspring for these two traits? This would be our little friend down here. What's the probability of these two parents having this offspring? Well, if, again, we count all the boxes, there's 16 of them. How many times do we see this potential genotype? Only once. So it's a 1 in 16 possibility. Okay. Now, what proportion of the offspring are likely to have? So let's actually write that down real quick. So this is a 1 in 16. Okay, we got 1 in 16 for recessive, homozygous recessive. What is the proportion of offspring that are likely to have the phenotype for dominant yellow and recessive for shape, for wrinkled seeds? Well, we see that there are 3 here. All right, there's 3 wrinkled seeds that are yellow, so that's going to be 3 out of 16. Now, what's the proportion of offspring that are likely to have the phenotype for round and recessive? So it would be round and green. Well, we see 1 here, and 1 here. So that's, again, 3 out of 16. Okay, and that leaves us with 1, 2, 3, 4, 5, 6, 7, 8, 9 that are yellow and round. So that's 9 out of 16. Okay. Now I mentioned before the 9-3-3-1 ratio, right? So when we're talking about dihybrid crosses, independent assortment tells us that we have a probability of 9-3-3-1 in terms of outcomes with a dihybrid cross. Okay, so this Punnett square gives us this ratio, 9-3-3-1. Okay, now these are all over 16, but this is the ratio here, 9-3-3-1. Okay. All right, now what evidence did Mendel have that the alleles segregated independently in the cross that we have here? Well, he actually found all of these combinations of phenotypes in his results. So that's how he was able to know that the alleles actually segregate independently. So, in other words, the yellow and the wrinkle doesn't sort together, or we'd get completely different results. They have to independently assort in order for us to get the results that we do. So independent assortment is defined here on screen. It says genes for different traits can segregate independently during gamete formation. All right, so Mendel's principles of heredity, which he observed through patterns of inheritance, they really do form the basis of our modern understanding of genetics. So, inheritance is determined by units called genes, which are passed from parents to offspring. This is really a summary of what Mendel was able to figure out. He also figured out that where there's more than one form of a gene for a single trait that exists, some of the alleles may be dominant and others might be recessive. What he also figured out was that each adult has two copies of each gene. Each one has one from each, and each one gets one from each parent. So these genes segregate from each other when gametes are formed. And then the alleles for the different genes usually segregate independently of each other. And so those are the four statements that summarize Mendel's findings, and they really do provide the foundation for our modern understanding of genetics.